Okay, dear audience, my name is Jack Quetzels. I'm a nephrologist from the Radboud University Nijmegen in the Netherlands, and I welcome you to this webinar of today. And we are very pleased that Professor Orson Moe is able to give this webinar. He was asked at the latest moment, but he was so nice and kind to accept the invitation. Professor Moe studied at the University of Toronto and also completed his residency in internal medicine and nephrology there. And thereafter, he did practice research in physiological research at the University of Texas, and he is now a professor of internal medicine and physiology in the Center of Mineral Metabolism and Clinical Research at the University of Texas Southwestern, and director of the O'Brien Kidney Research Center. Professor Moe, it's a pleasure to have you, and I'm sure we will have a nice overview on the topic of uric acid disorders. Thank you.
Thanks, Jack, and thanks the organization for inviting me. It's a real pleasure. So I was given a fairly broad topic, uric acid disorders, which means I can do anything I want. So I'll try to make this informative. And okay, a little stuck here in the slide. All right. This is Dallas. Last count about 7.5 million people, so it's a medium-sized city. And I work in the medical school just shown here. Backdrop is downtown Dallas. And nephrologist by training and physiologist. So I see patients, I do research, and I teach in general housekeeping of kidney. So this is mandatory from our institution. I have to disclose everything. I have done consultation for the industry, these entities in the last 5 years, and I got paid for it. But I will not be talking about their products. I do hold patents in some products, and again, I will not be talking about any of those. So I'll tell you 3 short stories. Uric acid biology, transport, and some diseases in the second half. So uric acid contains 4 nitrogens, okay? And it's used for a lot of things. And here we're comparing ammonia that has 1 nitrogen, urea has 2, and uric acid has 4 nitrogens. And let's look across the animal kingdom, going from fish to mammals. And we're going to look at 3 functions: nitrogen excretion, acid-base balance, and osmoregulation. So ammonia is used for all 3 of these. Very few mammals actually use ammonia to excrete nitrogen. So this is not blank. There's a little bit of that. But we use ammonia for acid-base excretion, basically. Urea is used for nitrogen excretion and osmoregulation, really across the phylum. Uric acid is used for nitrogen excretion, acid-base balance, and also osmoregulation. And these functions are known as, if an organism uses ammonia to excrete nitrogen, they will be called a monotelic. If urea, like us, we use primarily urea to excrete nitrogen, we are ureotelics. And some organisms are uricotelics. Now, notice over here, there's a blank. So in mammals, we actually don't know what uric acid is used for. We have it, but we have no known function. Let's look at the plasma levels. Down here in birds and reptiles, they are uricotelics, so they use uric acid to excrete nitrogen. So the plasma levels are fairly high because that's what they make to excrete nitrogen. Well, in mammals, we don't use uric acid to excrete nitrogen. We are primarily ureotelics. But a strange thing happened. In higher primates, our plasma levels are almost the same as birds. You say, what's going on? We don't use uric acid to excrete nitrogen. Why are we competing with the bird and the reptiles in terms of plasma levels? If you look at fractional excretion, Okay. Now, in the reptiles and birds, they use uric acid to excrete nitrogen. It's filtered and secreted, so the fraction excretion is very high. In mammals, the fraction excretion is very low because we don't synthesize that much urate. But look at the higher primates. Not only do we have high levels in the blood, we have very low fractional excretion. It looks like the kidney is deliberately hanging on to uric. So there's usually a reason for that. And this will be evident in a minute. If you look at the gene uricase, this is what is used to metabolize uric acid to the allantoic acid. There are cumulative mutations in uricase, in the promoter, in exons. By the time we get to humans, there's no chance for this gene to be expressed. We have a gene of uricase. It's called a pseudogene because it's never expressed. And that's the reason. Over evolution, over these 40 million years, we have managed to accumulate all these inactivating mutations of uricase to make sure we don't destroy uric acid. So silencing of uricase happened during hominid evolution. So the uric acid levels are low in prosimians, very much like the other mammals, and it gradually increases, becoming very high in human. This completely parallels the loss of uricase activity. So the first thing that happens, we are not destroying the uric acid, we want to keep it. Now, if this was an accident or many accidents, the kidney would have reacted and excreted, but we see the opposite. Okay? As the uric acid level goes up due to the loss of hepatic uricase, the urine excretion goes down. So this cannot be an accident. The kidney is working in conjunction with the liver to make sure the plasma levels go up. So we are ureotelics carrying high uric acid levels in the blood. Why? Is it because we want to have disease? Well, evolution never really deliberately get us into trouble. We actually don't know why we keep high uric acids. There are at least 4 theories, and I don't have time to go into them. These 4 theories may be true, may not even be mutually exclusive, but they provide explanations as to why the higher primates elect to maintain a high uric acid level in the blood. That's a little bit about biology. So let's look at how we move uric acid around. 3 major organs: the liver, the gut, and the kidney. So we eat some uric acid, but very little. In fact, we eat purines that in the liver gets converted to uric acid. People say about a third, maybe 30, 40% is secreted into the gut. And that's probably by and large true, but hasn't been measured that well. In the gut, we have bacteria that contains uricase. So they eat the uric acid. It's probably about 20 plus species of bacteria that are uricase positive. So they feed on the uric acid. For that reason, you should never measure uric acid in the stool because that does not reflect what we secrete. It reflects what we secrete minus what the bacteria has eaten, and it's actually very low in the gut. There are old studies where radioactive uric acid was infused into the blood, and then the radioactivity is traced in the gut, and that's where these numbers came from. Majority of the excretion is by the kidney. Okay. About 60 to 70% of the produced uric acid ends up in the urine. So things have to move. Okay, we have a lot of transporters that are supposed to take uric acid. And I'll caution you in this field as you read the literature. Every time you get a list of transporters, don't try to read this. This is meant to show you that many of them. I'll also go through a list of criteria. First of all, they usually discover in vitro in a heterologous system, like they inject the cDNA into xenopus oocytes, and they see if these oocytes will take up uric acid. And then they need to be localized in the right segment. For instance, this is in the segment in the proximal tubule in the kidney, where you have a lot of uric acid transport. So they transport uric acid in the laboratory when expressed in oocytes. They are in the right place. Now we make a deletion in a mouse. And if the mouse expressed the appropriate phenotype of uric acid disturbance, and we have a story. Caution here is uric acid handling in rodents can be quite different than humans. From some of these transporters we have identified, they are actually sitting in different places in the nephron when you compare a rodent to a human. So rodent data alone, take a little caution. And then the next step is usually some sort of a human GWAS, a genome-wide association study. What this is a very powerful statistical association of allelic variants in a particular gene product, let's say one of these uric acid transporters. See if they correlate, beg your pardon, see if they correlate with uric acid levels. And a lot of these candidates have survived up to 4. But only 3, and I'll show you which 3, have the final proof, which is human Mendelian mutations. These are single gene monogenic defects with inactivating mutations. And then there's a uric acid phenotype. So if all these 5 are satisfied, then this is a true uric acid transporter. So in the kidney, it's handled by 3 mechanisms. It's filtered, it is secreted, and also reabsorbed. Kind of complicated. Okay? People often ask, why is this so complicated? It looks like secretion and reabsorption is working against each other. It's kind of stupid. You carry a package up the stairs, you carry it down, you carry it up again. And I think this is because we did not invent our nephron. We inherited it from earlier species. Okay, and remember, lower vertebrates, non-mammals, are uricotelics. So they have fractional excretion that's 5 or 10. So they filter and secrete. So we have some remnants of the nephron from a reptile or a bird. Some of the transporters are still present, but we are primarily reabsorbing the filter urate. The normal fractional excretion is somewhere between 5 to 10%. Remember, in the bird, it's 500% or 1,000%. So these are remnants of earlier form of the proximal tubule that we have just kept. So these are the players. It's a very complicated slide, and it's not meant for you to memorize, but I classify them into the apical membrane in the kidney facing the lumen. They are all in the proximal tubule in humans. And you have the basolateral transporters. And on the left, I have the ones that participate in secretion. There's one that looks like a uric acid channel. There's a sodium coupled transporter. And there's an ATPase that directly couples uric transport to ATP hydrolysis. On the right are the absorbers that take uric acid from the urine back to the blood. And they're all coupled transporters. In other words, we're coupling sodium organic acid transport with organic acid urate transport. When you put them together, the organic anion recycles itself, and you basically have created a sodium coupled urate uptake mechanism. So they use different combination and manage to reach the same end result. These 3 are the rock stars because they have the most information of the list that I showed you earlier. So it looks like it's some sort of a painting you see in the museum, but it's actually the histology of uric acid crystals deposited into soft tissue. It's bad news. On the right is the polarizing image of that. But I could frame this, put it in my office. Now, let's look at some diseases. Most of us are physicians, and we deal with disease. These are primarily crystallopathies. Whether uric acid in solution can lead to disease is not known. It may. A lot of people believe that it may. But the documented human diseases are all diseases of crystals precipitating. And let me organize them for you. Always nice to organize things so you can remember. You either have problems with too much uric acid crystallizing in the extracellular space, or you have problem with crystals forming in the kidney, either in the interstitium or in the urinary space. And these can be congenital or acquired. It's a good way of thinking about it. The congenital conditions are rare. Most of us may not see a case. Okay? And you can further divide them into metabolism, transport, or unclear, maybe transport. Okay? And you can further break them up into inheritance patterns: autosomal dominant, recessive, or X-linked. Disturbance in the mechanism example will be xanthinuria, which is an inactivating mutation of xanthine oxidase. It's like a lifelong consumption of allopurinol for buxostat. Okay? They have low serum uric acid, low urine uric acid, low fractional excretion. So it's not really a uric acid disease. But backing up on the metabolism, xanthine does not get metabolized, and they can form xanthine stones. Look very much like uric acid stones. You have to be, I tell the trainees, first of all, it's very rare, so if you diagnose that, you'll be a hero. Everything looks like uric acid stones except you don't see uric acid in the urine, it's very low. Then go xanthine. Rare. You can also have monogenic enzyme deficiencies that leads to increase in uric acid in all compartments. This is because during purine metabolism, when you have these enzyme deficiency, and this one here is an activating mutation, you have too much of the substrate going down the uric acid pathway. This is not that rare. It's apparently fairly high in prevalence in Japan. It's hereditary renal hypouricemia because you leak uric acid into the urine. I've seen only one patient in my lifetime, so it's unusual. But in Japan, they apparently see it fairly often. And then you've got this familial juvenile hyperuricemic nephropathy, or due to a number of different mutations, can be uromodulin, a TAM-Hos4 protein, hepatic growth factor. In rodents, the renin gene has been described to lead to this. The mechanism is unclear. It's believed to be possibility of transport, but they get high uric acid and they get renal failure. So these are rare congenital diseases. I want to talk a little about URAT1. So this is transmembrane protein, is a transporter that takes uric acid. Okay? It's in the apical membrane of the proximal tubule. It's in the right place. In GWAS, these genome-wide association studies, it's been linked to serum uric acid level in multiple places in the world, in Europe, in Asia, in the US. So it's probably true. Okay? GWAS can lead to artifacts, but these were very reproducible studies, and I think that's going to be true. But still, it's an association. Okay? But we have the final proof, which is this disease of renal hypouricemia. The serum uric acid levels are actually quite low. Instead of hundreds of micromoles, they are tens of micromoles. FEU rate can be very high. So when you see a low blood level, high urine level, you think of renal hypouricemia. They're leaking. Okay? And a lot of the patients remain undiagnosed. So I think one of the reasons why this disease is rare In Europe and the US, it's because we don't diagnose it. If they get hematuria, they may get some attention. If they just have crystals, no one ever think of crystalluria as a disease. So I believe there are people walking around with renal hypoglycemia and activations of URAT1 and never reach diagnosis unless they have the following. Okay? They can form stones. Even if they do that, the one case I saw had uric acid stones, and the doctor thought this is the garden variety case of uric acid urolithiasis. Don't worry about it. Put you in case citrate, you'll be fine. The patient was not fine. Okay? And that led to something else, that led to something else, that led to something else into this diagnosis. This is when everybody gets medical attention. They get exercise-induced AKI. It's a form of acute urate nephropathy, like you would see in the tumor-lysis syndrome. They have a massive load of urate over a very small period of time, and the kidneys shut down. So in Japan, a lot of these people will have the first diagnosis from an intensive afternoon of some sporting event, and then they get hematuria, and the next day they're very sick. They show up in the emergency room. They have acute kidney injury. Okay? So if you don't have these two conditions or hematuria, I think a lot of people just go through life without a diagnosis. And here's an example. This is an interesting case report, again, came out of Japan. What happened here was a kidney donor, a living donor, who had mutations and was never diagnosed. So this is in the beginning, this program, the protocol biopsy, it's a male recipient, so that's the Y chromosome, the red dots. And with time, some of the stem cells from, I'm sorry, this is the donor kidney, so it's the X chromosome, the woman with asymptomatic mutation donated the kidney. Sorry about the mistake. And with time, the recipient, who was a male, cells started infiltrating the kidney and populate some of the cells with Y chromosome. So what they saw here was right after transplant, the serum uric acid dropped to a very low level, and uric acid increased. And then with time, their theory is as the recipient cells populate the kidney, and I don't know how much that happens, this partially got better. Okay? And they did sequence the donor who was heterozygote carrier of a mutation of URAT. So it just shows you that the disease can be very mild, particularly when you're heterozygote. Okay. Now, this has become a very hot target for pharmacological therapy. I think you all know of these drugs: probenecid, sulfentrazone, benzpronone, and lesinuride. These are inhibitors of URAT1 and promote uricosuric. Therefore, they lower serum uric acid. Okay, I checked this recently, and these are the drugs in the pipeline. No kidding. Okay, this is serious. Every company is jumping on the bandwagon to get inhibitors of URAT1 so you can use it to lower serum uric acid. And this will be an interesting development. You all know the surprising outcome of SGLT2 inhibitors. Initially, we thought, oh, you get some glucose out in the urine, get better glycemic control. But no, it makes everything better. Even a non-diabetic patient with kidney disease, if you go on SGLT2 inhibitor, you do better. I should take some myself. You should put in the drinking water. Okay? And I draw your attention to this paper written by Sanjay Nigam and University of California, San Diego. And Sanjay talks about he made a deletion of URAT1 in the kidney, and he found a lot of metabolic pathways being altered using an omic approach. And this approach is when you change one thing, a lot of secondary and tertiary events happen and can lead to unpredicted results. I think we're witnessing that in inhibiting glucose entry to the proximal tubule. We think glucose entry goes in, it goes back into the blood. That's all it does. It's a conduit. It's a transit at an airport. No, when you inhibit that, a lot of things happen, and they were surprises, shocking surprises. So as people get put on URAT inhibitors in the next couple of years, I'll be very curious to see what happens to things that are not uric acid related. Just planting a little seed in your head so you can think about it. Now we go to the acquired disorders, which are common. These are the three well-described ones: gout, urate nephropathy, acute, and kidney stones. Okay? And let me just talk a little bit about kidney stones. It hurts, we know that. This is one of our trainees, Emmanuel, is a nephrology fellow. He wrote this really nice article that came out in seminars. Emmanuel basically classified them to bystander, where uric acid is just hanging around, got captured by a proton, and you get stones. Instigator, uric acid pairs with sodium, and sodium urate promotes calcium oxalate precipitation. So you get calcium oxalate stones. And it can be the actual perpetrator. That means urate get on with ammonium and precipitate and form an ammonium urate stone. Three types of stone that involve uric acid. That's a uric acid molecule, and it dissociates. One of the hydrogen comes off of a pKa, the association constant of 5.4, which means low pH it will want to be uric acid, high pH it will want to be urate. Difference is urate is very soluble, uric acid is not. So in this diagram, we plotted urinary pH against total uric acid and undissociated uric acid. So this is uric acid plus urate. At pH 6.5, you can have very high uric acid, and you're still under the solubility limit of undissociated uric acid, which is rather low. But as long as your pH is 6.5, you're probably safe, because none of us will have this much uric acid in the urine. However, if you sit around with a pH of 5.5, you don't need a lot of uric acid to precipitate. And this is what's happening with uric acid stones. It's a disease of urine pH. It is not really a disease of uric acid. It is truly the innocent bystander. Okay? The pathophysiology is complicated. I'll summarize it for you. It's a multi-hit model. Okay? You start out in the gut, and you end up in the kidney. This increased organic production by complex mechanisms having something to do with the microbiome. There's an increase in organic acid uptake and impair hepatic metabolism and also increased production. You have decreased renal excretion of ammonium. As you know, ammonia, NH3, it's what buffers the proton. Okay? If you don't buffer the proton will get onto whoever will take it. The urate, that's quite high in human urine, gets that proton and it precipitates. And this is basically a disease of the metabolic syndrome. It has to do with fairly complex things, and I don't have time today to go through it. So when you see the kidney stone, the uric acid stone is only the tip of the iceberg. This is what's happening in the organism. Okay? It's a mismatch of evolution and our lifestyle. It's with a lot of things. Metabolic syndrome, a lot of hypertension is a mismatch between what we have evolved to be and what we've chosen to be. Patients sometimes don't believe that, but I tell them you are forming kidney stones because of that. Now, the second condition has to do with sodium urate. This is the different forms of uric acid. And in real life, we never see the divalent form, but we do see the monovalent form. And this is the solubility. Uric acid solubility goes up as pH goes up, but quite opposite, sodium and K urate, it goes down. Okay? Luckily, we don't have a whole lot of sodium, K, and ammonium urate, but some of us have high sodium in the urine. We eat a lot of purines, and the urine may be a little bit alkaline, and then you're going to have ammonium urate precipitation. So it acts on calcium oxalate by 3 mechanisms. It's a chemical effect of salting out the calcium oxalate. That's a direct effect of sodium urate. It binds to a lot of the colloidal inhibitors of crystallization, because sodium urate exists also in a colloid phase. And in the crystalline phase, it looks like a calcium oxalate crystal. So heterogeneous enucleation, epitaxy. So it fools the calcium oxalate into precipitation. So this is a disease of high sodium urate in the urine, causing calcium stones, also acronym of HUCU, hyperuricosuric calcium erythrolysis. And the last condition is due to urinary tract infection. This is urea. As you break down urea, you release ammonia, and the carbonate raises urine pH and increases the urate concentration. The uric acid now becomes all urate. And these two start dating and get married. And then if you eat a high purine load, that also helps in raising the urate. So they get ammonium urate precipitation. Very difficult disease to treat. This is what gives you these staghorn calculi. You have to get rid of the infection. So let me summarize. We're just about right time, getting close to 35 minutes. There's a conflict between evolution and lifestyle. I have not talked about why we end up with high uric acid. I gave you examples of 4 models. So if we truly get a beneficial effect from uric acid, we are now pushing it too high, putting it at risk of crystallization in soft tissue, in the urine, and in joint space. It's a human disease of uric acid crystals. We're still working on why. This cannot be an accident because it was very steady, very steadfast. It's going through the different higher primates. Uric acid has to have some sort of benefit. So these models have been proposed. They have some merit to that. Nothing has been proven. We treat the metabolism and the transport, and one needs to know what we are doing. People are jumping on to lowering serum uric acid in people who don't have gout. I'm not sure that's justified. Maybe it is. Some subscribe to the fact that high uric acid by itself per se can accelerate the progression of CKD. That has some indirect proof but never been proven. It's a lot of people to treat unless we know by lowering uric acid, we slow down CKD progression. But it's a big number. In the United States, CKD 3 or above, it's close to 20% of the population. Imagine the market share if you have a drug that 30% of the population should take. So some of this is not pure. It's driven a little bit by product. But I show you that cartoon of the number of inhibitors of URAT. Unbelievable. If you have a drug company, you try to make one. And something I've not talked about is the microbe and host interaction. We are host to a lot of uric acid eating microbes because we have high serum level you secrete into the gut. You basically have an open buffet. Come, everybody, come eat this stuff. It's free. And the microbe is actually interacting with the host. One of the things I talked about, the microbe paying us back. Thanks for the uric acid. It was delicious. I don't have a credit card. I'll give you some fatty acid in return. It's a great source of calorie, not knowing that we are already calorie loaded. And it's changing the mucosa in the gut. So that's also a very interesting topic. Can't fit this into this talk. So I believe that's all I have. And I go back to the slide that was given to me to end the meeting. And this is the one. Be happy to take any questions.
Okay, thank you very much, Dr. Moe. And we are ready for the questions, and already some questions came in, and maybe to start with two ones. First question is, you are questioning what would be the possible role for these high uric acid levels in humans, and I wonder, from these Japanese patients where they have this mutation and have low levels of serum uric acid, is there any information if these patients have on the long term some unknown problems?
Yeah, excellent question. Excellent question. So the answer is no. So as you know, these patients, they have actually a normal lifespan. So some of them are basically in the 80s, 90s. The things you look for would be increase in cardiovascular disease. Some people believe that uric acid maintains cardiovascular integrity. And there's some data in humans that infusion of uric acid can increase blood pressure transiently. Well, these individuals with renal hypouricemia are completely normal. The only disease they have is exercise-induced AKI, and that's bad. So if they're careful, they have no phenotype, which makes this very, very difficult. The experiment of nature has been done, and the results have been negative.
Okay, well, thanks. And the second question is, so you have discussed this stone formation. Do you think that in patients with elevated levels of uric acid, that the condition which was formerly called urate nephropathy, does it exist?
Yeah, so this is referring to a condition that's been discussed for now a long time. When I was in medical school, which was a long, long, long time ago in the 70s, I don't think any one of you at that age, maybe some of you are, based on the original Framingham study, uric acid was considered as a risk factor for cardiovascular disease. So when I was in medical school, people were prescribing allopurinol to lower uric acid, and that became disproven. Now it's CKD. So chronic urate nephropathy is very difficult to prove. Acute, nobody argues. Chronic urate nephropathy is extremely difficult to prove because every now and then you'll find uric acid crystals in a biopsy in a local area of acidity, and the fact that a lot of CKD patients have a slightly high serum uric acid, it precipitates. So it's extremely difficult to say, okay, this is due to that. They may be true unrelated. There are intervention trials that has been, part of them are positive with very small effect. A lot of them are negative. The take CKD patients, look at the slope gradient and look at the slope of eGFR, and then put them on something, allopurinol or febuxostat, any drug, and they look for a change. And they're either negative or very small difference. I believe in China, it is actually approved to slow down CKD. Maybe there you can take some population studies to see if it works. It will not help us if everybody goes on it. Some people have to miss it. So today, as we speak, I would say the clinical data to support the existence of urate nephropathy is weak. I will not say it does not exist. And a lot of people, including myself, I try to do this in rodents. Well, rodents are used to very, very low uric acid levels. If you bring them to the human level, they get sick. Is that human urate nephropathy? I'm not sure. If I raise a mouse uric acid to human levels, they get deposits in a lot of tissues. And the kidney actually has a lesion that's more similar to acute urate nephropathy than the human chronic urate nephropathy. So I'm sorry this is a long answer, but I would not say I can rule it out, but the evidence for its existence is weak. And patients ask me that. I'm sure your patients ask me that. Doctor, my uric acid is just borderline high. I think we need to lower it because this will slow down the progression. I usually say there's no evidence, but sometimes they want it anyway.
Yeah. Okay,
And,
thanks.
Yeah.
Then the questions from the audience is one question is if we can give losartan or sodium SGLT2 inhibitors in patients with, well, urate nephropathy. Maybe we should consider high urinary uric acid excretion.
So losartan will affect. You could use these drugs, and I just have to one word of caution: high uric acid, okay, hyperuricemia in the absence of kidney stones may not be a disease. I think if you're using those drugs for another reason, you get a bonus from reducing the urinary uric acid. So if I have a higher than normal uric acid in the 24-hour urine, I don't get stones, I'm not sure what I'm treating. Okay? So just a little word of caution there. Yes, they work.
Yeah, so I assume that if a patient with CKD is not having kidney stone disease, you are not measuring regularly uric acid excretion in the urine.
Not routinely, but we do every now and then to look for the other parameters. The urine sodium, the calcium, they become very low before you even have clinically detectable D deficiency. Also look at oxalate, because oxalate nephropathy, I believe in contrast, do exist, and it's a very shy diagnosis. So we don't do it routinely, but in some patients we do 24-hour urines to look at metabolic parameters. That's also a conditional answer.
Okay. Then the next question is if you can tell something about the renal outcomes in patients who are treated with uric acid lowering medications.
So this is what I alluded to earlier. In some, so a lot of these studies, the renal outcome was not a primary, so it becomes sort of an ad hoc type of analysis. But studies have been done using routine xanthine oxidase inhibitors, and the results are mixed. Some studies have found a slight improvement in the slope of decline of eGFR. Some have found nothing. No studies have found worse outcome, so it's either no effect or very small effect. It's mixed.
Okay, thank you. Let me see. Other questions are. One question is whether the stone composition or urine chemistry really helps, provided that there are such chemical interactions at the molecular levels. While a predominant calcium oxalate stone diagnosis point to anything, or it's a misguiding thing, as same can happen with uric acid calciuric states.
Let me see if I understand the question. Are you questioning whether the stone composition helps?
Well, I assume that the question relates to the fact that you showed probably these interactions with uric acid binding to ammonia and giving rise to calcium oxalate stone formation, and whether then in this complex chemistry of the urine, whether the measurement of these electrolytes can help really to tell something about.
Let me see. Let me see. I assume I understand the question. If I don't, please ask again. The composition of the stone helps because this condition of the hyperuricosuric calcium oxalate stones always have some uric acid or urate in the stone because it's the process of heterogeneous enucleation. If you see a stone that's purely calcium oxalate, it's very unlikely to be precipitated by uric acid. That's number one. Number two, in the 24-hour urine, very often you see normal calcium, normal oxalate, normal citrate, all the risk factors that lead to calcium oxalate stone formation. Okay, the three big ones: hypercalciuria, hyperoxaluria, hypocitraturia. Instead, you see high urine uric acid. Look at it carefully. The urine pH, it's making more urate than uric acid. And very often you also see high urinary sodium. In that case, sodium urate will precipitate. And when it does, if you have borderline chemistry for calcium and oxalate, that will come out. So it's not just the stone composition, but the whole picture of urinary chemistry and stone composition. You look at the mild nature of the calcium stone risk factor, but you see really quite robust hyperuricosuriia. Then the way to attack that stone former is to not to treat the calcium, oxalate, or citrate, but lower the urate. Usually the first line we use xanthine oxidase inhibition. And when that's the case, very often you will see success. I have to say, if I see a lot of calcium oxalate stone formers have some hyperuricosuriia. If the stone has no urate in it, and they have other identifiable risk factors for calcium stones, such as hypercalciuria, if you treat that uric acid, often nothing happens. The really ideal pristine patients, the one I told you about, stone composition supporting the mixture, and very mild calcium chemistry. Okay.
Okay.
I hope that answered the question.
Yeah. Another question is whether these uricothelic animals make stones and have uric acid deposits in the tissue.
Okay. They don't. So unless they are really overwhelmed, okay, they don't form stones. Look at a bird. Okay. I'm sure you have the experience where you're sitting outside having lunch on a beautiful summer day and some white stuff ends up on your sandwich from an unwelcome visitor flying above you. Okay. That white stuff is very high in uric acid. Why is that? Because the avian kidney actually precipitates uric acid. That's the whole reason, not the whole reason, a good reason why you use uric acid to excrete nitrogen. First of all, it carries 4. Second, it precipitates in the urine. So you don't need a large urine volume to hold it because the bird doesn't really drink that much. So you excrete the uric acid as crystals, but then it forms at the very end of the nephron. It's discharged into the gut. So it does precipitate. You want it to precipitate. In the blood, in the tissue, in the joints, it does not. Okay, and that's still a little bit of a mystery. There must be inhibitors. The serum levels, I can understand. They're not much higher than us or some of your patients with high. But in the tissue, I would expect a bird or a snake to have uric acid deposition, but it doesn't happen. But it could happen if you overwhelm it. If you load a bird or a chicken with a purine load, and this has been described in veterinarian literature, from contaminated feeds with very high purine. The chickens, they die. What did they die from? From a massive total body uric acid deposition as a disease. But in physiology, they don't. Okay, and I'm suspecting there are inhibitors. We can find those inhibitors. We can give it to humans. Very good question. Yes, very good question.
Okay. Then it's one question on CKD and a question whether you would consider to have a target level of uric acid in patients with CKD.
No. So I answered a little bit of that earlier.
Yeah.
There's no target. There's no target level because we don't know what's at risk for the kidney disease. I treat everybody who has gout attacks, and a lot of them do. But asymptomatic hyperuricemia, I usually tell the patient the data is mixed. I'm not comfortable to expose you to the drug. I mean, we do see about 1 or 2 cases of Stephen Johnson a year from patient allopurinol. I mean, it's not. And then CKD, as you all know, the incidence of interstitial nephritis is higher than the general population. So I don't. And I'm not telling others what to do, because since there's no data, I told you in China they do that because they believe the literature is strong enough to support slowing down of CKD. So they use the clinical cutoffs and they treat it when it's higher than the reference range. I don't.
Okay,
The patient
thanks.
has gout, I do,
Yeah, yeah. One other question is about what is the relation between uric acid and fructose?
Yeah, the metabolism of fructose can be a lot of things. Uric acid's one of them. So in very high fructose consumption, you get hyperuricemia, you get hyperuricosuriia. So one of the theories, the condition that you probably don't see in Europe, is this Mesoamerican nephropathy seen in a lot of the farmers in Central South America and Mexico. I live on the border state, so I do see occasionally our friends across the border. It's a combination of different things. These workers are constantly working in the sun. The urine osmolarity is maximal. They consume beverages. These are Mexican beverages that are loaded with fructose. They are super, super high in fructose. And they are thirsty all day and they drink 5, 7, 10 bottles of that. And they have high uric acid. I'm not sure that's causing the nephropathy, but when we refer these patients to, hey, there's high uric acid in the blood and high creatinine, and they related, we do work them up for the high uric acid, and it's coming from the fructose. So I don't know if someone has a slightly above normal uric acid in the blood is a problem. But fructose is converted to uric acid. Is that a risk factor for CKD? I don't know. I asked those patients, I tasted these beverages. It's impossible to drink it. It's so sweet, but it's very popular in Mexico.
Okay. And then your opinion about the hypothesis that uric acid is involved in the development of hypertension.
Okay, so it's not my hypothesis, but very quickly summarize the data for you. There's a statistical association of blood pressure and uric acid levels, not just dichotomous, but the level. Okay, there you cannot conclude anything on causality. There is some data looking at patients before and after getting allopurinol for other things, but those patients also got a lot of medical attention. The blood pressure is lower. Very difficult. Okay? I think if you just see a doctor, the doctor tells you to exercise, eat better, your blood pressure will come down. Mm-hmm. Very difficult. The last one is this example I cited before. Studies were done mostly in young, healthy people. Acute infusion of sodium urate salt gave rise to a transient rise in blood pressure. And the most convincing study was done in children of hypertension. The reason for that is the organs are relatively preserved. If I get hypertension, I probably have some degree of vascular disease, stiff vessels, my heart may be a little below LVH. Children, their organs are pristine. So the idea is whatever is causing the primary hypertension, it's probably alone. They lower the uric acid with the usual therapy. They did it by a xanthine oxidase inhibition. They did it by uricosuric, and there was a lowering of blood pressure. So of all the studies I mentioned, there are tons of them. That's probably the most convincing one. So if a person has hypertension and high serum uric acid, one could try. I don't think it's wrong to try. It's not asymptomatic hyperuricemia. This is hyperuricemia without gout, but blood pressure is a little bit high. So if you ask me, I'm on several drugs. I'm on thiazide beta blocker. ACE inhibitors, blood pressure is still submaximal. I would say, hey, why don't we lower the uric acid and see what happens? I think starting everybody on xanthine oxidase, everybody with hypertension and high uric acid may be going a little bit too aggressive, but I think there's a role. If you look at that, I just summarize 100 papers for you, but the last one, which are these children who had the uric acid lowered, the blood pressure came down. Okay? I was impressed, came down. So judge for yourself.
Yeah, okay. So I think what is clear from your presentation is that it's a very interesting molecule, and I'm sure that in the coming years we will get more information about its role in various diseases. So I thank you very much for this beautiful presentation. We are now coming to the end. It was a success, and there were not only questions but many remarks on the quality of your presentation. So thank you very much for giving the presentation today. And
thanks for inviting me.
Yes, and I would like to give attention for the audience to the coming webinars which are on the screen on the genetic of stone disease by the end of March, and then in April we will have webinars on IgA nephropathy and on STAC-associated hemolytic uremic syndrome. So again, thank you very much, Professor Moe. I thank the audience for attending the meeting, and we see you back next time. Thank you very much.
Okay, thank you.
Bye-bye.
Bye.